This video is going to cover sections 5.7 and 5.8 from your textbook, which is how genetic variability is created by sexual reproduction and the basics of meiosis. Genetic information generally is packaged in chromosomes, and in eukaryotes it usually occurs in pairs. If you take all the chromosomes from an individual, an organism, and you line them up like you see over here in the right-hand side, this is what you'd call a karyotype. So you can probably guess this is a human. You've got 22 what we call autosomes, and then one pair of sex chromosomes over here. So this whole thing is called a karyotype. Humans have 23 pairs, right? 22 that we said are autosomes, and then 2 that are sex chromosomes. The 2 chromosomes that make up the pair are called homologous chromosomes. So like these 2 right here would be homologous chromosomes of chromosome number 3. Okay, we sometimes call them homologs. Cells with pairs of homologous chromosomes are called diploids. So if you have 2 copies of your chromosomes like humans do, you would be diploid. If you have 3 copies, you'd be triploid. 4 copies, tetraploid. And if you have just 1 copy, which we'll see here pretty soon, those are called haploid. So what if chromosomes aren't paired in the cells that just have a single copy of the chromosome? Those types of cells are called haploid if you've got only 1 copy of the chromosome. So for instance, in your body, any reproductive cells that are produced by diploid organisms like you would contain half of that number, and those would be haploid. Okay, so your reproductive cells have only 1 copy of each of your chromosomes rather than 2. Organisms that have more than 2 copies of each homologous chromosome are called polyploid. So like I was saying, you could have triploid organisms that have 3 copies, tetraploid, which would have 4. Some plants have lots of polyploidy. For instance, strawberries are octoploid. So as you might guess, that means that they have 8 copies of every chromosome. So they've got quite a few copies of just the individual chromosomes. This is a YouTube video that won't let me play on this recorder for some reason, but we will probably do this lab when I get back. This lab is extracting DNA from a strawberry. I'll show you how to do that. Let's talk about N, which is usually italicized in lowercase. So every species has a different number of chromosomes. Right? Humans have 46. Other species have varying numbers. The number of different types of chromosomes in a species is its haploid number, and we call that number the N value. So humans, even though they have 46, they only have 23 unique chromosomes minus the sex chromosome, the pair of sex chromosomes. But we'd say the human N value is 23. You have 23 unique pairs of chromosomes. Okay, so 23 individual chromosomes. So we have 2 copies of 23 different chromosomes, therefore our N value is 23. Most of the cells in your body are diploid, or what we would say is 2N, right? 2 times N, okay, which equals 46. If you were triploid, it'd be 3N. Tetraploid would be 4N. Octoploid, like the strawberries we talked about just a moment ago, would be 8N. So this just acts as a multiplier depending on how many copies of the chromosomes that species has. I said these terms a few minutes ago, but here they are officially, so make sure you get these into your notes. Autosomes, these are the pairs of chromosomes with nearly identical DNA sequences that are found in diploid cells of both sexes. So these are the non-sex chromosomes. You have 46 chromosomes in total. 44 of them are autosomes. Okay, the sex chromosomes are the chromosomes that determine the individual's sex. Humans have 2 of them. Okay, so a lot more autosomes than sex chromosomes in a human, right? There would be 22 pairs of autosomes and 1 pair of sex chromosomes. In mammals, okay, usually, so like you and I, right, mammals, 2 X chromosomes indicates female, and an X chromosome and a Y chromosome gives you males. Those would be the sex chromosomes for mammals. Other species use different inheritance patterns. Birds are different, right? They have the opposite type of inheritance. So the sex chromosomes can vary depending on the species. They're not always X and Y. Here's an example of 2 different karyotypes. You can see that, right, they've got them paired up here. So this one's got, pair of chromosome 1, the homologous pair of chromosome 2, same thing over here. The only difference that you'll note is on the female one, you will see 2 X chromosomes. They're very large chromosomes, relatively speaking here. So these are both quite big. And a male karyotype, you'll see an X chromosome and a Y chromosome, which is much smaller than an X. So if I give you a picture and say in a future quiz and say, what type or what's the sex of this individual, you want to look for these sex chromosomes to see do they have an XX or an XY to determine that. On your chromosomes, obviously, is your genetic information, and the genetic information codes for genes, which are going to be little segments along the chromosome. So a gene is a sequence of nucleotides found at a specific location on a chromosome that encodes information for building functional molecules or proteins. So the information that's stored on here helps the cell to build proteins. Okay. The slightly different versions of genes are called alleles. We're going to see a lot of this when we talk about inheritance in our next unit, but alleles. Okay, so you might have the gene for hair color, right? Might be located here, but there might be different alleles like brown hair, black hair, red hair, blonde hair. Okay, there might be those are the different alleles that could be at those locations. Okay, so if this is a pair of homologous chromosomes, you might have one allele here for blonde hair and one over here for brown hair. I'm just using hair as an example. It's probably not exactly how it really goes, but you get the idea, right? So these would be on opposite chromosomes here. These are pairs, but you might have two different alleles. They might code for the same thing. This one might be brown, this one might be brown, or maybe one's brown and one's blonde. But those are the different, alleles. We've seen this term before, but let's get a definition down again for you in your notes. Mutations. These are changes in DNA that can occur when cells either make mistakes when copying their DNA or when the DNA gets damaged by some sort of factor like UV light or harmful chemical exposure. If a mutation that gives rise to an allele that happens to occur in the cells that produce reproductive cells, that allele might be inherited by offspring and subsequently passed on from one generation to the next. So the only way that you can pass on these mutations is if they happen in your reproductive cells, right? Most mutations are, and this is a future point here for sure, you might see this on quiz or test or the final. Most mutations are harmless. Most of them you don't notice. Most of them are inconsequential. They don't affect you positively or negatively. Occasionally they can be destructive or lethal, so they might be harmful or kill you. Rarely do you get beneficial mutations, right? Evolution's had a long time to work out its process, and it's resulted in humans looking like they currently do, so it's not going to be very often that you're going to get a beneficial mutation that hasn't arisen before. So it's pretty rare to get beneficial mutations. Okay, so most of them are harmless. Some can be harmful or deadly. Very rarely would you get a beneficial mutation. Okay, diving a little bit into meiosis here. We had mitosis at the end of our last unit. And now we're going to go into meiosis. So meiotic cell division or meiosis, this is when a diploid cell, so you're starting it with 2N, ultimately gives rise to haploid daughter cells that each contain one member of each pair of the homologous chromosomes. So you're going from 2N down to 1N. Eventually, those two individual cells, one from the female, one from the male, are going to fuse together. Each one will be 1N. They're going to fuse together and form a zygote, which is a fertilized egg, right? And this becomes a diploid cell that grows and develops on into a new human. Meiosis is going to produce 4 haploid cells at the end of its process. So you should probably know that it makes 4 haploid cells at the end of the process. It's different in males and females. In males, you get 4 identical haploid cells. We'll talk about what happens with female cell, with egg cell meiosis, because you only get 1 that's actually viable, and you get 3 what we call polar bodies, which we'll see later here. But you end up with 4 cells regardless, okay? So meiosis is split into 2 parts, meiosis 1 and meiosis 2. Meiosis 1 is where you have separation of the homologous pairs of chromosomes into 2 daughter nuclei, and each daughter nucleus receives 1 homolog from each pair. So they split the homologous pairs of chromosomes. In meiosis 2, the sister chromatids are going to separate into independent chromosomes, and they get distributed. They just distribute each chromosome to a daughter nucleus. So it's going to split twice here. So meiosis 1 and then meiosis 2. Here's a nice diagram of how that happens. So you're starting with the original cell here. It is going to split the homologous pairs, okay, in meiosis 1, and then meiosis 2 happens, okay, and you end up with sister chromatids of each homolog separating into independent chromosomes. So we end up with, you can see here, each one got 1 chromosome. So we're going down from 2 copies of it down to 1 copy of it, and as you can see, we ended up with 4 cells being produced. Okay, so here's what it looks like in picture form, right? Meiosis 1, it's going to have the same steps that mitosis had. We just call it, instead of, prophase, metaphase, anaphase, and telophase, we're going to call it prophase 1, metaphase 1, anaphase 1, and telophase 1. And we're going to take a look at each of those individually here in just a moment. Okay, in meiosis 1, you start with prophase 1. So prophase 1, you have homologous chromosomes. They pair up and they exchange DNA. You get this phenomenon over here called crossing overs, which we'll see here, which is a source of genetic variation. So maternal and paternal homologous chromosomes line up together. So this is be like, say, chromosome 1. Okay, so you have, your 2 in, so you have 2 copies of chromosome 1. They line up together, and you might have points where they cross over, like you see over here. And those points are called chiasmata. So those are the cut ins, so when they cut off here of homologous chromosomes that are now binding maternal and paternal chromosomes. So this point here would be what we call the chiasmata. And then what happens is they actually exchange. You can see they exchange parts of their chromosomes. So this chromosome that, say, might have been originally dad's, and this one might have been mom's for you. Instead of having one that is 100% dad and 100% mom that might get passed on, now you've got one that is, 90% dad, 10% mom, and this one's 90% mom and 10% dad. So they're not 100% the same copy that you received from your parents. There is some amount of crossing over that has happened, and that happens during prophase 1. Okay, I would say make sure you know where does crossing over happen. It happens in prophase 1 when the homologous pairs, when they pair up. Other than that, prophase 1 is also going to have the same thing happen like we saw with mitosis. You're going to see the nuclear envelope breaking down, the spindle fibers form, and those spindle fibers are going to attach at the kinetochore. So that's really very similar to that prophase that we saw back during mitosis. Just that we're attaching now to the homologous chromosomes. Metaphase 1, the homologous pairs are going to line up, like you can see here. They are assorted randomly. So remember, like you might have one that was mom's chromosome and one that was dad's chromosome. Well, it's not as if like all the mom's chromosomes line up on this side and all of dad's go on this side. It is random. This one might be mom and dad. This might be dad then mom, mom and dad. Who knows? Okay, it is random, and since you're human, right, you've got 23 pairs here. So that's kind of a lot of degrees of randomness here as to how many are going in each direction when this thing splits up. This random assortment has a name. It is called independent assortment, and I would say make sure you know that independent assortment happens during metaphase 1. Okay, so you get this random alignment here of maternal and paternal chromosomes. You don't know which one's going in which direction, and that's going to, again, increase genetic diversity. And then you get anaphase 1, where the homologous pairs separate. Okay, so they will separate here. And then telophase 1, the spindle fibers disappear, the cytokinesis starts to happen, right? You get the formation of 2 new cells. The nuclear envelopes might reform, however, usually they don't because, and this is another good future question here, at the end of telophase 1, these cells are going to go straight into meiosis 2 and do this process over again. So they don't wait around usually. They usually go right on to meiosis 2. Okay, now meiosis 2, I'm going to go ahead and put a big old star next to this because I promise you're going to see this as a question somehow. Meiosis 2 is functionally the same process as mitosis. Okay, it's still going to be prophase, metaphase, anaphase, and telophase, but we're going to call it prophase 2, metaphase 2, anaphase 2, telophase 2. And it's functionally the same process as mitosis. Okay, that's certainly a future question. So let's break this down and see what this looks like. Okay, so we end up with 2 cells at the end of meiosis 1 after the homologous chromosomes have separated, and those, are then going to have to get pulled apart. So the homologous chromosomes have been pulled apart, and now the chromosomes themselves are going to be separated into individual ones, which you can see what's going on here. So prophase 2 looks just the same, right? You've got spindle fibers forming. Metaphase 2, they line up, along the metaphase plate. Anaphase 2, they get pulled apart. Telophase, you start to see the cells pinching off, the reformation of the nucleus, the nuclear envelope, and then you end up with, at the end here, 4 haploid cells. This is another YouTube video which I'm not able to watch while I'm recording, So we will watch this once again when we are together in class.